Hello, 11th grade. So this is the second lesson in our unit on statistics, the IA, and experimental design. And we're going to be talking about the next phase of design. After you make sure your study is ethical and safe, what is the next thing you need to be thinking about? And that would be, the part of that is variables. So you learned about this a long time for many years. We're going to go over the basics and talk about how you work with more complicated variable situations and how you pick good constants because we're still developing that skill set in this course. So you have to make sure that you have very clearly identified variables in any experimental design. It's a key component. If you can't identify a variable for a question you have, then your question does not work. So that's a really good way to eliminate ideas maybe you have if you can't figure out how to make it specific enough to a certain independent and dependent variables, then it's not a good choice. So quick reminder, the independent variable is what you were changing or manipulating. So in this really simple plant experiment example, the independent variable is the liquid being used to water the plant. So change the liquid, they have Coke sometimes, juice sometimes, water sometimes, different liquids to water the plant. The dependent variable is what you're measuring to see how it depends on the independent variable. So in this one, they're using the height of the plant as their dependent variable. And then constants, sometimes there's something called control variables. That's not a good idea at this level because we're going to be talking about control groups and experimental groups. So we're going to talk about control variables and control groups. It gets really confusing. So the way that we talk about this now in science is we say constants, things you're going to keep constant, and we talk about control groups. So please use the term constants, and those are the things you're keeping constant to make sure nothing else affects the dependent variable besides the independent variable. So in this plant experiment, it would be the type of plant we're being used, the size of the pot, the amount of liquid, the soil type, anything that's going to affect the growth of the height of the plant that is not the liquid. Okay, so a good way to make your IA more appropriate to the IB level is to have multiple dependent variables. Now you should always stick with only one independent variable, and you might change the magnitude of it or give kind of different iterations. Like for example, this one we have the independent variable is the liquid used, but they have different liquids, right? They have Coke and juice and water, different ones. That gives you some variety and some better information, but it's still one independent variable. But you can measure multiple dependent variables. So what else could you measure for this one besides just the height of the plant? Well, you could also measure the root size of the plant, the biomass of the plant. You could measure the number of leaves, the leaf photosynthetic surface area. You can do a lot of different things. How healthy it is. All those kinds of things are different things you can measure, and the reason why it's good to have multiple dependent variables in biological studies is because often things are not as easily predicted as we think. Okay, so just because we think that we're going to see a height difference doesn't mean we are. Maybe in that particular plant species, if it gets more sugar in its water source, its roots are going to really change, but not its shoot. So that's why it's really good to have multiple dependents, and you can always choose to focus on one or two in your analysis, but collecting data on multiple gives you a chance to find the best and clearest pattern between your independent and dependent variables. Be really careful though if you're going to choose to do more than one dependent variable that you do separate statistical analysis for each one, because just because the roots are changing doesn't mean the shoot is changing. They're separate things. How each one's related to the independent variable. When you change your independent variable, like I just said earlier, it should be different iterations. So iteration means the same thing. Oops. Went too far. Went really too far this time. Okay, so the iterations means that you're changing something about it, but it's still the same independent variable. So for example, if your question was, how does height affect the number of three-pointers scored on a basketball court? You would have people of a variety of heights to look at. Or how does age affect ventilation rate? A variety of ages. Those are iterations. Those are different levels to look at. But it's still the same independent variable. So be careful with that one. Let's talk about constants. So it's really important that you don't forget any key constants. You remember all the constants that are important for your experiment, but you also avoid unnecessary constants. And this is something that students often do when they're first developing their skills in experimental design, is they tend to either make really simple constants in lower grades, 9th grade, 10th grade, and then in upper grades, 11 and 12, I find my students often go too far and they list so many constants, some of which are unnecessary. And the IBO does not like that. They will punish you if you have unnecessary constants. So it's a really balancing point of having sufficient constants to make sure your independent variable is the only thing affecting your dependent variable, but you're not adding extraneous unnecessary constants either. So how do you figure out which ones are necessary and which ones are not? Two things: experience. So as you do labs and develop those skills, which you've been trying to do a lot of stuff this year to help you with that, that'll help. And then research. So look at similar studies. What did they keep constant? What was not necessary to be kept constant? That kind of stuff. What did they not do? So in this experiment, let's look at this simple plant growth experiment. Which constants are necessary and which ones might be unnecessary? So the list that they have here, they have the type of plant, pot size, amount of liquid, the soil type, and they have etc. So if there's anything else, that would be there, or should be there. Well, if the independent variable is the type of liquid, then you should definitely have the same volume or the amount of liquids. They have that one, that's good. I would say the frequency of that liquid being given. So are they getting 500 mils of liquid every day? Is it every other day? That kind of thing. The time of day can affect how liquid is absorbed by plants based on transpiration rates. The size of the pot, if it's something growing tall that's flimsy stem, should it all have a support to grow up against? It's a good idea with plants or things that are growing, but you start at a constant level or you do percent change. So either give all plants start from a seed the same day, or the plants all start at like 5 grams size, small plants, something like this. Something to keep that constant. Do you need to keep constant the number of leaves on each plant? Do you need to keep constant the initial height of each plant? Not necessarily. If you're going to do percent change, which is the best way to show growth usually, that doesn't have to be kept constant. The number of leaves, that's really hard to control, and if it's hard to keep it constant without damaging the specimen, it's probably just not a good choice to do. Some people might talk about keeping it constant as they put it in the same environment, so maybe they have them all in a greenhouse, and they talk about, okay, so then I have constant temperature, constant wind exposure, constant light. If you're keeping it in the same environment, all those things are kept constant anyway, and you don't need to repeat them. Okay, so always, the best way to make sure constant is necessary or not is think back to your dependent variable. Will that factor affect it? If you're keeping constant, so for example, if you're keeping the timing of watering a plant constant, will that affect the height of the plant? Well, we know water is necessary for photosynthesis, which is how plants get energy to grow. And if they're not getting it at a time when they can photosynthesize, will they lose it? If we put it all on the surface of the soil at the beginning of the day or the afternoon, will the sunlight evaporate quickly? This is something you can look at. If you're not sure, then you can look at a little bit of research and see if that's the case or not. If you know for sure it's going to affect your independent variable, or you think it might, then you have to keep it constant. If it's not going to affect it, like the color of flower pot it's in, then don't worry about it and don't include it. It's unnecessary. And in scientific studies, they always want you to be concise as much as possible. Okay, so when we're planning experiments, you have to kind of keep in mind the data you're going to end up with in the end. And this is a big difference between a beginner investigation, a 9th grade, 10th grade investigation, and what we'd expect from an IB student. It's both the quality of the data and the quantity of the data. If you look at the IA rubric I posted for you in the unit resources folder, you'll see some of the points you get for accuracy and quality and quantity of data. So your data needs to be both valid and reliable. So when you're designing your methodology, your kind of general idea of how you will conduct your experiment, you should be keeping in mind at this point in your brainstorming process, can you do that experiment with enough validity and reliability to get an accurate number? So validity, remember, is does your test measure what you want it to? So if you look at this image here, these are all invalid. The target is measuring, let's say we'll go back to one, it's measuring plant height, how it's affected by different liquids. And the only one that's actually measuring that is this one. So how do you know that the liquids are what's affecting the plant height? Does your test measure what you want it to? And reliability, how consistent is your measure and is it repeatable? So if you have all your data points are fairly similar, not a lot or no outliers, then that's going to be reliable. But if it's actually reliable and invalid, then it's going to be measuring what you think it's supposed to be measuring and consistently measuring that. So if someone else, can they repeat your experiment? Are your data points similar? How big is the standard deviation? That kind of stuff. So how do we make sure that we are planning a valid experiment? Well, the big thing for this is constants. Anything that can influence your findings should be kept constant. So only the independent variable changes and affects the dependent variable. That's a weird typo. Okay, that's better. Okay. A good way to do this too is if you have a control group and an experimental group. So in a control group, you're not including the independent variable. So you can see if there's something that's affecting the results besides the independent variable. So this is really important in studies with biology, especially because there's so many factors that can affect organisms. So for example, in this little example here, we have a medication we're testing to see if it affects the response, some sort of response. Let's say it's a blood pressure medication. We want to see how it affects the blood pressure in these individuals. So group A, the experimental group, will be given that blood pressure medication. And then we track their blood pressure and see if it changes or not. It's the dependent variable is the blood pressure, how it's changed. Maybe the percent change in blood pressure. The control group is going to get a placebo. They're not going to get the independent variable. But everything else is kept constant in the two groups. And then we see what their response is. So for example, let's say in this kind of experiment, you're looking at blood pressure, maybe you're going to keep constant diet, amount of exercise, hours of sleep, stress level, these kinds of things that can affect blood pressure, age, gender, all those kinds of things. So that would be the same for both of them. The only difference here is this one's getting the medication, is getting just a placebo, the inactive medication. And then you can see if both groups' blood pressure decreases, then something else is affecting the blood pressure besides the medication. There's something else causing this change-dependent variable besides the independent. So what control groups are really important, especially to ensure validity in biological experiments. So here's another example. So I have a control group. The question is, will students' test scores be affected by distracting sounds in the testing environment? So here's my control, here's my experimental. Think for a minute, how is this necessary? How does this work? Well, I want to see what's going to affect their scores, their testing scores, so I have to give them both the same test, same amount of time, same kind of students, but people are really complicated, and how they do on tests is not just caused by one thing. So I need to have a control group, see how they're going to score as a group without any noise to compare the group with noise. So a good way to think of it is that the control group is identical to the experimental group in all ways except for the absence of the experimental condition or the independent variable, which in this case is the distracting sounds. Okay, so let's do a little practice and experiment here. So we have Joe, and Joe wants to determine how the increasing amounts of growth hormones affect the mass of pea plants. So he's already narrowed down his independent variable is the amount of growth hormone, and independent variable is the mass of the plants. So how can he make sure his experiment is valid? Take a minute and think about this. Well, Joe should be using constants to make sure his experiment is valid, that he's actually measuring what he thinks he's measuring. So he should keep the amount of water the same, he should have the same plant size or plant age, the same amount of fertilizer, the same amount of sunlight exposure and intensity of the sunlight they're exposed to. Make sure there's no predation or parasitism on the plants. Make sure the growth period is the same if you want to see how their mass is changing. Where it's really hard to control things like plant size and age, The good thing to do is change your dependent variable to, instead of being just the mass of the plant, be the percent change of mass to see how much it went up from where it was before. You should have control experimental groups. The control group should have no hormones, but everything else should be the same for them, whereas the experimental group will have different amounts of hormones. So that's one way that Joe can make sure his experiment is valid for this particular inquiry here, investigation. Okay, to make sure it's reliable, that's the other part here. The best way to make sure it's reliable is to do many, many trials. 5 is the minimum we're going to say for IAs. 3 is what you've probably been told for a long time. And the reason why we say 5 now is because 5 is a minimum for standard deviations from the mean. And that's a very basic test you need to be able to do for statistics, for graphing, all those kinds of things. So at least 5 trials. Remember, a trial means exactly the same thing multiple times. Okay, another way you can do increased reliability is you can have many different individuals. 30 is the minimum for most times you work with individuals like animals, people, that kind of organisms. Because there's so many variables about them that you just can't control. You can't keep constant about them. Think back to our ventilation practical 6 lab. It's really hard to keep all those factors constant. So a good way to do that is to have lots and lots of data of 30 individuals, each one doing 5 trials. Means you're going to have sufficient data that any outliers shouldn't mess up your conclusions, and you'll really see that what you're finding is consistent and reliable. The last thing to do is to make sure that your procedure is really, really clearly written, so your exact investigation, your exact experiment can be replicated by another scientist, see if they find the same thing or not. So let's go back to Joe's experiment here. So how could he make his experiment reliable? Well, first thing, group size. He included at least 50 plants in each group. 30 control plants and 30 experimental plants. He can repeat the entire experiment 5 or more times for a total of at least 150 control plants and 150 experimental plants. Now, if he doesn't want to have that many plants going at once, because it's a lot of plants, right? He can do 30 plants each, but do the period of growth maybe for a week, and then restart and do a week 5 times. So instead of doing it all in a short period of time, you can use the same plants over 5 weeks. That's another option he can do to have lots of trials. And the procedure, he can write a very detailed procedure so his experiment is repeatable by anybody looking to check the reliability of his data. Okay, a lot of times in biological investigations, it's really tricky to make sure everything's reliable and valid. So because we're working with living things, it makes everything way more interesting, in my opinion, but also a lot more complicated. Right? You can't keep everything controlled all the time, and unpredictable things happen in the lab all the time. We can't keep organisms constant or controlled particularly well. So what often happens is we do pilot experiments. A pilot experiment is where you're trying to check that your original methodology works. Check for any kind of major errors, maybe in the environment, maybe the species you've chosen, any of those things, and gives you a chance to improve your methodology. So we're going to look at some examples of some IAs next class, and one of them did a pilot experiment, and I'll talk to you guys about how that works. This is really important if you're trying to figure out which species to use and you're not sure if the species you've chosen of plant, whatever it happens to be, is the right one to use. Pilot experiments are a great way to do that. Okay, so your task today is to take your 10 possible ideas you already made and narrow it down to the 5 best ideas. So you can take those 10 ideas and narrow it down to 5 ideas that you can do virtually. So if you want to do an experiment with plants in your home, that's wonderful. Do it. Go for it. It's a great idea. But if you don't want to do something like that at home or working with people over Zoom or something, then you need to find something you can do a different way. With a simulation, with data that already exists, that kind of thing. And remember, you can always put a star beside ideas you want to do in the lab to be your real IA you do in August. So for each of those 5 ideas that could be your potential mock IA, you're going to figure out what are the variables, constants, the control and experimental groups, as well as is there a pilot experiment necessary for your experiment or not. That's your task. Okay, so your homework is to upload those notes. Make sure it's a split page notes again. So you have your notes on this what looks like on one side and your notes for your particular IA on the right-hand side. And make sure for each of those 5 ideas you include your IV, DV, constants, control experimental groups, and pilot experiment necessary or not necessary. Okay, so next class we're looking at some example IAs and talking about the research we need to do before we begin actually conducting our experiment. Okay, hope this is helpful for you guys, and hope everyone's staying home and safe. Have a good day.